Hi, I'm Evan Kelemen, and I'll be talking to you today about some work we've been doing in the Rehan lab on a prosocial bee in the genus Ceratina. Bees in the genus Ceratina, or small carpenter bees, are an interesting genus to focus on because it is a cosmopolitan genus occurring on every continent except Antarctica, and it is speciose with more than 200 species. This genus contains social and non-social species, as well as species that exhibit social and solitary phenotypes. Bees in this species are long-lived and loyal to the stems that they nest in, allowing for detailed observations to be done and for nest transplanting. Extensive work in this genus has been done focusing on the maternal care within species. Mothers guard nests from parasites and groom their offspring, as well as mothers feed larval and adult offspring. Work in the Rehan lab has also focused on understanding how this genus moved across the globe. Early biogeography, using 2 nuclear genes and 1 mitochondrial gene, discovered that the likely origin of this genus is in Africa about 47 million years ago, and it's from here that they rapidly radiated around the globe. Ceratina dispersed 4 times out of Africa in the Eocene and early Oligocene, and then dispersed 3 more times between the Miocene and the present. Interestingly, there is no back dispersal back to Africa. This may be due to niche preemption, where it's easier to disperse into an area with an empty niche than into one where the species already belongs to a functional group that is there. Also, at the continental level, it suggests that the composition of bee fauna is more strongly influenced by very early dispersal events rather than later physical barriers to dispersal. As mentioned before, Ceratina is socially variable, and we tend to find that in the tropics, these species are bivoltine, so they have 2 broods per season, and they tend to be facultatively social. Whereas in temperate regions, these species tend to be univoltine and solitary. This pattern holds true when tested. The ancestral state reconstruction, mapping biogeography, and social behavior show a very strong correlation between these 2 traits. So we can learn more about these species by comparing how the environment influences these tropical social species and these temperate solitary species. So we did that first by looking at Ceratina australensis. So this is a tropical species known to be bivoltine and social. Within a population of Ceratina australensis, about 13% of them are social. Now these nests are considered semi-social, so it's made by a pair of full sisters, with one sister being in the primary egg layer and the other sister doing more of a worker-like behavior of foraging and guarding. To investigate how the environment influences the social behavior exhibited by Ceratina australensis, we sampled this species across its range in Australia. And we looked at things such as temperature, because this influences social behavior in ground nesting bees, and also precipitation, because it's been shown to influence behavior exhibited by stem nesting sweat bees. We also looked at the parasitism rate in this population, because that's been suggested to be a selective pressure pushing species towards sociality. And we also looked at maternal investment, because how much a mother invests in her offspring can influence the social environment exhibited by a species. We looked at these sites across multiple years, and temperature varied between sites and between years, and similarly, precipitation varied between sites and between years. We found that there was no difference in the percent of the population that exhibited the social phenotype between these sites and between years. However, we did find that during hot years, mothers produce smaller offspring, suggesting she invests less in her offspring during hot, dry years. Interestingly, social nests did produce larger females, so they were able to invest more into their offspring. We found that nests were typically female biased. Interestingly, we found that South Australia had a more even sex ratio. And very few studies have compared the sex allocation of stem nesting bees across a population and climate zone, but existing research suggests that female biased sex ratios are conserved in bees. And this unbiased sex ratio of nests in South Australia is actually pretty unusual. We also found that there was high variation in parasitic pressure across site and year. So why do we find both solitary and social phenotypes within a population. It may have to do with the costs and benefit of sociality. Long-term studies have found that solitary individuals have higher fitness than social individuals. However, previous work on Ceratina australensis has shown that the low frequency of social nesting in this species is a risk avoidance strategy in response to a variable offspring survivalship, likely due to parasitism intensity. And as we have shown in the study I showed here today, parasitism rate is extremely variable. So the benefit of sociality in this species may be that there is always somebody to guard the nest while someone else is out foraging. This unpredictability of parasitism may maintain the social phenotype, so altruism in a volatile world. This idea is supported by modeling work, which has shown that the variability of parasitism creates years where it's beneficial to be social. And so if there's a high frequency of these high parasitic years, it's enough to maintain a social phenotype within the population. We can compare these findings to how the environment influences a temperate species, Ceratina calcarata, which is univoltine and solitary. Ceratina calcarata exhibits subsocial behavior. There is prolonged maternal care, where the mother feeds both the larval offspring as well as the adult offspring. In fact, this species shows 2/3 of the hallmarks of eusociality. There's overlapping generations, with the mother and daughter living within the nest. There's reproductive division of labor, because the mother produced what's known as the dwarf eldest daughter, or the Cinderella daughter, in which she produces a smaller sized daughter in the first brood cell, and this daughter emerges early and will actually take care of the other adults within the nest later in the season. However, there is no cooperative brood care, as the mother is the only one that takes care of the daughters as brood. Interestingly, a potential constraint on the species that prevents eusocial colony formation is the fact that the species needs to disperse in the spring. A little bit more on the life cycle of Ceratina. So they overwinter in their nest as adults. And then early spring, they'll emerge and mate. And then the female will go out and found a nest. And so she'll dig a hole into a stem. And throughout this, she'll be collecting pollen and nectar, so foraging to provide provisions for her brood, making pollen balls and then laying eggs onto these pollen balls. And then from here, the workers or the daughters will emerge and stay in the nest while the mother will continue to go out and forage and feed them so they can overwinter as adults. And in the case when the first daughter is fed less and made smaller, she'll as well go out and forage and help supplement the adult's diet so they can overwinter. Now, to test how the environment affects Ceratina calcarata, similar to what was done in australensis. Populations were sampled across their range. The temperature and precipitation experienced by bees at these sites differed. Now, interestingly, the species exhibits a stable social structure across its range. Even though the season is longer in the south, in this Georgia population, bees in this population aren't exhibiting bivoltinism, so we're not seeing multiple active brood stages. Instead, we're just seeing brood spend longer as mature brood or as adults. So we've looked deeper into how the environmental factors affect Ceratina calcarata, particularly how it affects their size overall. So we've looked at across their range and through time, so dating back to 1902 to present, seeing how differences in environment have affected them as environments have changed due to climate change and increased urbanization. And so we combine these morphological measurements on these samples with things such as average summer temperature and land use data through time to see how these factors are influencing them. So we found that temperature and agriculture actually exert opposing pressures on the species. So in populations that experience increasing temperatures, these bees become smaller, and so this is likely to do with biochemical processes, such that individuals that develop at warmer temperatures are smaller. So these are a physiological constraint. But then we also see that populations that develop in areas that have higher agricultural cover tend to be larger. And so this might have to do with these individuals having to be larger to disperse across this matrix of agricultural crops where they cannot find food amongst these crops, and actually having to look further and explore further to get the food they need to survive. Now we have examined both a tropical species, Ceratina australensis, which is social, and a temperate species, Ceratina calcarata, which is solitary. So now we can leverage this information to gain further insights. So interestingly, the results examining both Ceratina australensis and Ceratina calcarata has shown us that the behaviors exhibited by these species are ecologically stable. In australensis, we've seen that social polymorphisms exist despite differences in temperature, precipitation, latitude, and parasitic pressure. And this suggests that there may be a genetic component to these social behaviors. This provides an excellent opportunity to compare the genomes of these species, especially since genomes for both of these species have been sequenced and annotated. Analysis using these genomes are underway, including investigating genetic differences among populations previously examined in this talk, as well as understanding how differences extend to the microbiome of these species, as well as examining genes underlying behavioral differences among these populations. I look forward to telling you about these results in conferences to come. So this work was done by a lot of individuals, and so I'd like to thank everybody in the Reedham Lab. So every current and past member, as well as the many collaborators that have helped this work come to fruition. So I'd like to thank all of them. And I'd like to thank you for taking the time to listen to this talk. Thank you.